We're good. Okay, so I think we're, looks like most of us are here, if not joining. So I'll start the introduction. It's a really great pleasure to introduce Dr. Daniela Drummond Barbosa. Daniela is a professor in the Department of Biochemistry and Molecular Biology in the Bloomberg School of Public Health at Johns Hopkins University. She got a bachelor's in biochemistry and immunology in the Universidade Federal de Minas Gerais in Belo Horizonte in Brazil. I'm not sure if I said that quite right, but it sounds very beautiful. It sounds like a very beautiful place. Daniela got a PhD in genetics in the laboratory of Daniel Di Maio at Yale, where she studied the mitogenic signaling induced by viruses, in particular papillomavirus. She did a brief postdoc in the same lab and later did a postdoc in the laboratory of Alan Spradling at the Carnegie Institute, where she started to work in the Drosophila germline and its response to stem cells and differentiation to nutritional cues. She became an assistant professor in Vanderbilt University in 2002. And in 2009, she joined the Department of Biochemistry and Molecular Biology at Johns Hopkins, where she has been since as an independent investigator studying the pathways that link metabolism and the environment to stem cell differentiation using the Drosophila germline as a model system that she's going to tell you about. Daniela has received numerous awards and honors. I'm going to just cite a few, very few. In 2014, she was elected as a fellow of the American Association for the Advancement of Science. And in 2020, she became a member of the electorate nomination committee for that organization. She has also was a regular member in the Development and Differentiation in Cancer peer review committee from the American Cancer Association, where she was the vice chair and chair. She was also a regular member in the Cellular Mechanisms in Aging and Development study section from NIH. And she was recently asked to be the associate editor for the journal Genetics. At Johns Hopkins, she was also received multiple years the Excellent in Teaching Awards. So without any further ado, I want to thank Daniela for visiting with us today and for giving the seminar on her work. Thanks, Daniela.
Thank you, Francisco. I'm really enjoying my day here today. And if you guys have any questions about anything I'm talking about, you don't need to wait till the end. You could always unmute and ask me the question, and that would be totally fine. So Francisco already mentioned my areas of interest, and I hope you can still hear me because occasionally, apparently I freeze. But so what I thought I would do today is start with a brief introduction of the broad question that we're interested in and the Drosophila ovary system, and then walk you through the progression of my lab since I started it in 2002 all the way to now, and give you I'll summarize the data from each phase and then give you just snippets of examples of how we got some of those data, just so you can get a flavor of how things have moved on over time in our thinking and the questions that we are asking. So the broad question that we're interested in the lab is, how do stem cell lineages respond to the external and physiological environment in the context of multicellular organisms? So there are pools of stem cells in multiple tissues in our body and the bodies of other multicellular organisms. And those stem cells have intrinsic factors that control their behavior. They have local signaling that control, that interact with those intrinsic factors and control their maintenance versus differentiation modes. But the reality is that these organisms are in highly variable and always changing environments, both in terms of external factors. For example, if you think about various types of stress that can be in the environment, or differences in nutrient availability or oxygen tension, et cetera, but there are also changes that can occur at a physiological level just from the fact that organisms age, for example. So there's a huge number of factors that can affect what's circulating in your body. And we're interested in how those will impinge on the stem cells so that they can operate in the most optimal way for that particular situation that the organism is in. And we studied this question using the Drosophila ovary system. Part of it is just because the ovaries are so beautiful in Drosophila, but there's also practical reasons, the power of genetics in Drosophila, but also the fact that in the case of Drosophila, the production of eggs is sustained throughout adult life of the females from small populations of stem cells that are at the tips of each of the subunits of the ovary that are called ovarioles. And the ovary production is very sensitive to lots of environmental and physiological factors. So if you look at one of those ovarioles, then there's usually 15 to 20 ovarioles in each of the ovaries. What you see is a very well-organized assembly line of follicles that are in different stages of development. And if you look at one of these follicles in a cross section, it's kind of like a watermelon where you have on the inside the germline cyst, which is composed of 16 cells that are all interconnected by cytoplasmic bridges. And one of those cells becomes the oocyte, and the other 15 are nerve cells. And these cells have very different fates and very different functions. The nerve cells have a supporting role for the oocyte, but at the end of oogenesis, they squeeze everything to the oocyte and their nuclei undergo apoptosis. And surrounding the 16 cell cyst, there's a monolayer of follicle cells to complete the follicle. And these follicle cells have many signaling roles back and forth with the germline to help pattern the oocyte in the future embryo. And the very last role of the follicle cells is to secrete the eggshell. And once the egg ovulates, the follicle cells are gone. But all of the cells are required for the oocyte to develop properly. And as I mentioned, there are populations of stem cells that maintain egg production. So if you look at the anterior-most structure in the ovariole called the germarium, there are usually 2 or 3 germline stem cells at the very anterior of the germarium. And these germline stem cells are physically attached to a group of cells called cap cells, which are the major niche component. So they have an important adhesion role, and the cap cells and some other cells that are part of the niche secrete important signals that repress differentiation so that they can maintain the stem cell fate. And each time this germline stem cell divides, it self-renews, and it gives rise to a cystoblast daughter cell that's destined for differentiation. But the first step is for the cystoblast to divide synchronously 4 times to form 2, 4, 8, and 16 cell cysts. And then the 16 cell cyst becomes enveloped by the follicle cells that come from two follicle stem cells per germarium that are not labeled here. And then once this follicle is completely surrounded and formed, it will bud off. The germline cyst is surrounded, it will bud off as a new egg chamber that then progresses through oogenesis. And I should point out that if you look at this slide, you see these white colored stages here in the later stages. Those are vitellogenic stages where the oocyte is taking up yolk. And that starts at about this point in oogenesis when the oocyte is actively taking up yolk and growing very fast. And as you can imagine, this whole process is super expensive for the flies. And so when I started my postdoc in Alan's lab, I was in Alan Spreading's lab, there were 2 points of oogenesis control by diet that were well observed by everyone who worked with ovaries, which was if you had flies on a high yeast diet and they love to eat yeast, you would see all stages of oogenesis progressing nicely. But if you withdrew yeast completely from their diet, then there would be a retention of these mature oocytes. So because of a blocking ovulation, they would hold up these eggs, waiting for a better opportunity to lay them. And instead of investing in filling up these follicles with yolk, they would instead, this early vitellogenic stages would undergo apoptosis. So the flies basically cut their losses and try to invest on surviving and not like throw away resources. But when I started in Alan's lab, I was really interested in examining this response in more detail and looking at every stage in oogenesis. And I used the lineage tracing system that I'll mention later for a different experiment. But I was basically able to show that there are differences in how fast the germline stem cells divide and grow, as well as the follicle as a whole, and the follicle cells around the germline cyst change rates of proliferation, so that everything progresses faster on a yeast-rich diet compared to a yeast-poor diet. And in addition, if you remove the yeast from the diet, in addition to these points of control here, ovulation, vitellogenesis, I also discovered that there was a new point of increasing cell death in this early germline cysts that are about to be enveloped by follicle cells. And later on, as I started my lab, I realized if you withdrew the yeast for longer periods, even the fate of the stem cells would change and they would get lost from the niche, probably due to premature differentiation. So as a result of all these different processes, if you look at the numbers of eggs that the females lay per day, on average, for a healthy stock on a rich diet, each female can lay up to 80 eggs a day, which is a huge amount of mass because their eggs are pretty big. But if you remove the yeast from their diet, they quickly use all these processes here to slow down egg production drastically. And this is reversible, at least in the short term. And so ever since I finished my postdoc and started my own lab, we've been asking the same exact question. What are the physiological mechanisms mediating this response to diet? And more recently, we've expanded this question to think about additional inputs besides diet. So early on, when I started my lab, I focused on the most obvious signaling pathway that was screaming at us, which was insulin signaling. And I had already shown with global mutants in my postdoc that they essentially mimicked the deprivation of yeast. But once I started my lab, I wanted to understand, like, what is the role of insulin signaling itself directly, intrinsically in the ovary? And what we found that the major, most important source of insulin-like peptides among the ones that were known at the time were from the brain of the flies,
Are we don't have Daniela with us, right?
I do not see her in the
Okay. So she did say she had a connection that was a little spotty, and it was just freezing in with signal before, but the sound was still there. So let's just wait. There we go.
Oh,
Yep.
she's back.
Yeah.
You're muted, Daniela.
Sorry, I was saying that I froze, and then the Zoom was completely frozen. I had to force quit, and it took me a little while to figure out that, but sorry.
No
And I
Thank you.
had nightmares about this, so they were well-founded.
No problem. Thank you.
Anyway, so what we found was that insulin-like peptides were coming from the brain to control various steps of the germline response to diet. And early on in my lab, we also figured out that the maintenance of stem cells was also being controlled by insulin-like peptides. So I'll just show you like one brief example of how we got some of this data, just as a flavor. And without going into all the details of how we do genetic mosaic analysis, the essence of this approach is that we raise flies that are heterozygous for the insulin receptor in this case, because we wanted to understand at first, like what cells really require the insulin receptor to receive the signal and control the ovary, to find out if it was like a direct mechanism to the ovary. And then at a specific point in time, when the flies are already adults, we induce a few cells to become homozygous mutant for the insulin receptor, and they can be recognized by loss of a fluorescent marker. So over time, we can follow what happened to the cells derived from that original cell, which makes a mutant clone. And what we found, for example, when we made this mosaics, if you look at a control ovarian where both in this case, the fluorescent marker is beta-gal, being recognized with an antibody that has secondary with fluorescence. But so the beta-gal positive and negative cells in this mock mosaic, they all have a wild type copies of the insulin receptor. And you can see that normal progression of follicles, which stay in chronological order, which matches their developmental stage. But when we use two different alleles of the insulin receptor, when the germline cysts were homozygous mutant for the insulin receptor so that they couldn't receive the insulin signal from the environment, they grew significantly more slowly than the neighboring wild type egg chambers, as you can see in this break in the size sequence here. And we also expanded this analysis when we were analyzing a separate study, the target of rapamycin signaling pathway. And we found that TOR and insulin actually work together to control follicle growth. And just to give you an example here of the results we found with TOR, if we again mutate the target of rapamycin gene, which encodes a sensor of lots of different signals, including amino acids in the cells to control many different processes, including growth, what we see is that there is a real reduction in follicle growth again compared to the wild type neighbors. And if instead we make homozygous clones of a negative regulator of TOR so that there's too much TOR activity here, then you get overgrowth instead. And we showed that they were working together in the same pathway to control this. So that just gives you a flavor of how we got this results. And again, I already mentioned TOR, but we also looked at other roles of TOR in other stages of oogenesis. We looked at an energy sensor in AMP kinase. We looked at ecdysone, which is a steroid hormone in flies, and the homolog of receptor that's involved in sensing signals from fat cells in mammals. And all of them had intrinsic roles in the ovary, controlling like different combinations of those processes that are affected by diet. But the reality is that we don't have just the ovary receiving all the signals, right? The reality is that every organ in a multicellular organism's body has the ability to receive the inputs from the environment and from the physiology. And the question is, there has to be a refinement of how all these organs respond to these inputs through crosstalk between the organs, because they all have to know they're in the same organism, right? So there's all this potential arrow directions for communication when an organism is having a response to a physiological or environmental signal. And so how do you tease apart the contribution of each of these arrows, right? So we decided to focus first on this adipocyte to ovary arrow, because for several reasons, including even though I think all the organs are equally cool, we chose the adipose tissue first because, you know, even its most long known roles of just storing extra nutrients or extra energy and mobilizing it when there's not enough around, even that initially known role, it involves the tissue being nutrient sensing, right? Otherwise, how would it know when it's supposed to store or mobilize? But since those very old times, in the past 20 or 30 years, adipocytes have been really well recognized as having endocrine roles, as have many other organs now. Muscles, bones, lots of organs produce endocrine hormones, right? Or signals that can affect the whole organism. And as a testament to how important adipocytes are, when there are disruptions in adipocyte function, such as can be found in a subset of obese individuals, you have a lot of different problems, including a higher risk of infertility, cancers, other diseases, including like a worse outcome for COVID infection and all that. And in flies, the adipose tissue is organized differently because it's not its own organ. In flies, there is a fat body organ, which includes mostly adipocytes, but in the abdomen, at least, they are intercalated with inocytes, which have a liver-like function. And together, they also have immune functions. But this fat body is localized not just in the abdomen, actually throughout the flies, even in the legs, around the brain, thorax. And it has the similar roles to the mammalian adipose tissue. But in addition, it also has the endocrine roles during larval and adult stages. And even there's factors produced in the fat cells, in the fat body cells, that can control insulin secretion. So we thought we would focus on that first. So the first question we asked was, okay, if we have the entire organism having, plenty of nutrients, but somehow you perturb just the ability of the fat cells in the flies to sense amino acids or to have enough amino acids, would they be able to transmit that information to the ovary and cause a response there? So what we did is there is about 40 predicted amino acid transporters in flies. So a former postdoc in the lab, Alyssa Armstrong, she now has her own lab. So Alyssa decided to RNAi specifically in fat cells, one amino acid transporter at a time. And even then, the RNAi was not very effective. So it was like 60% effective. Because of course, we don't want to completely remove amino acids from any cells because they need it, right? So we just wanted a small, as physiological as possible perturbation so that we would just get a sensing, the consequence of sensing less, not really the consequence of absence of amino acids. And when Alyssa did this using a very specific driver that only affects only expresses in adipocytes to induce RNAi, what she found was that when she looked at first at the number of eggs per female per day, that at, doing this RNAi just in adult adipocytes. At day 6, there is not much of an effect yet, but from germline stem cell to an egg, it takes about 10 days in Drosophila, so that's not too surprising. But at day 12, she started seeing a significant reduction in the number of eggs that the females produced compared to the control. And this is only showing 3 amino acid transporters, but there were additional ones that had all consistent changes in egg production, and actually later analysis of the ovaries, they all looked pretty similar. So the question was like, what are the steps of oogenesis that are affected by this perturbation in amino acids sensing or levels in the adipocytes? So I won't have time to show you every single step, but what she showed is that amino acids basically, when the adipocytes sensed less amino acids because of the knockdown of one transporter at a time, there was a small block in ovulation and a much more robust, faster rate of germline stem cell the germline stem cell loss over time. And I'll show you the germline stem cell loss in a minute. And the most obvious pathway to test for mediating these effects of amino acids in adipocytes was the TOR pathway, again, because it's such a well-known amino acid sensor. But surprisingly, when we inhibited TOR signaling in adipocytes, all we got was a very, very strong block in ovulation and no effect whatsoever on the stem cell numbers. So then the question was, what is the amino acid sensing response in the fat cells that is responsible for this faster rate of germline stem cell loss when the adipocytes sense less amino acids? So the next pathway that came to mind once we went back to the literature and tried to think about what made the most sense was the amino acid response pathway that was originally described and discovered in yeast, because yeast can synthesize all the essential amino acids, right? They can make all the amino acids that they need. But if they can find certain amino acids, if they have plenty of amino acids in their environment, they don't really need to spend all the energy to make amino acids. They can just use them from the environment. So they only activate the biosynthesis of essential amino acids when some are missing, right? And the way that works is that this decrease in amino acids leads to a surplus of tRNAs that are not coupled to the amino acids. And these uncharged tRNAs activate GCN2 kinase, which phosphorylates EIF to alpha, which then causes a global repression in translation, since there is fewer amino acids available. But in yeast, it induces GCN4, which then induces biosynthetic enzymes for amino acids. But in multicellular organisms, there is instead ATF4, which is induced by the translation of ATF4, which has a different 5' UTR, is increased, similarly to what happens with GCN4. And at least in cell culture experiments, a lot of the downstream targets are stress-related genes. Because ATF4 is a transcriptional factor, it induces stress responses. But in rodents, there had been a study at the time that had shown in vivo that this pathway functions in the brain of rodents to mediate a food avoidance response if the rodents are given a diet that lacks an essential amino acid. So they avoid eating that diet because it lacks an essential amino acids. So we thought, well, if that can happen in the brain of a rodent to avoid a certain type of diet, maybe this pathway could be active in the fat cells to cause a reduction in stem cell number in the ovary. So we tested that, and there were two predictions, and I will only have time to show you the results of the second prediction. But the first prediction was that, okay, so normally you have normal amino acid levels, the tRNAs are all coupled, the pathway is off, and you get normal stem cells. There's reduced amino acids, and this pathway is responsive. Then the extra uncoupled tRNAs trigger the pathway, and there's a decrease in stem cells. So our prediction was that then if we had normal levels of amino acids, in other words, we're not knocking down the amino acid transporter in the fat cells, but instead, all we do is inhibit individual aminoacyl tRNA synthetases, like genetically with RNAi, basically specifically in the fat cells, then you artificially create the situation where there's uncoupled tRNAs, even though there's not a shortage of amino acid. And what we found was that we saw a decrease in germline stem cell, just like we saw for when we just knocked down the amino acid transporter. So the prediction was validated by the experiment. The second prediction, and I'll show you the result for that one, was that, okay, so if this increase in uncoupled tRNAs is really activating the pathway through GCN2 kinase to decrease the number of stem cells, then if you knock down the amino acid transporter in the adipocytes and simultaneously knock down GCN2, you should turn off the pathway and actually maintain the normal numbers of stem cell, even though the fat cells have lower levels of are sensing fewer amino acids. And this is exactly what we saw. I'll show it later in the talk what the stem cells look like at the microscope. But if you look at the number of stem cells per germarium, and remember, each female has 30 to 40 germaria, one per ovariole. So we're just showing per germaria, but this represents a lot of stem cells per female. So if you look at 0 and 10 days of RNAi, in the control RNAi fly, there is a small loss of stem cells, which happens normally. If you do the knockdown of the amino acid transporter, the rate of loss is higher. But if you simultaneously knock down the amino acid transporter and the GCN2 kinase, then there is no loss. And we checked, and the degree of knockdown is the same in all the conditions for the genes that are being targeted. So this really showed then that the amino acids, when they're reduction is sensed in adipocytes, that the TOR pathway mediates the retention of eggs, whereas the GCN2 pathway downstream of uncoupled tRNAs is responsible for the decrease in germline stem cell numbers. But all we did was create more question marks. And in addition to this work looking at amino acids in the fat, sensing of amino acids in the fat cell, Alyssa also looked at whether insulin signaling acting the fat cells could feedback, crosstalk to the ovary to further refine the ovary's own response to insulin-like peptides. And what she saw is that if she manipulated insulin signaling specifically in adipocytes, then she saw really specific effects on the ovary that were only partially overlapping with the direct effects. For example, insulin signaling from acting on the adipocytes control early cyst survival, but they don't have a direct role in early cyst survival. So it just shows you the complexity of how multiple organs can be responding to the same pathway, and then there's additional crosstalk between the organs. So because we kept generating more question marks, we also thought, let's try like a different approach to get at how the fat might be communicating with the ovary. And we took a proteomic approach, which in retrospect was a little naive to try to identify secreted factors, but I could go into that later, why it was naive. But, so we wonder, it could be secreted factors, signaling metabolites, mobilized nutrients, anything, right? So we took this proteomic approach. At the time, the best way to do quantitative proteomics that worked for us was using iTrack. I won't go into details, but so we had quadruplicates or flies that had been kept on a rich diet for 3 days and then maintained on a rich diet for overnight, 12 hours, versus another population of flies that had been treated the same exact way on a rich diet for 3 days and then switched to a poor diet just for 12 hours. And we looked at the differences in the proteome that were caused by just a 12-hour difference in the diet because we wanted to catch early changes. And we picked the 12 hours both because of my early studies had shown that the ovary is already reacting by 18 to 24 hours in terms of our readouts, which means the response was happening earlier. And if we look at just the morphology of fat cells shown here with the lipid droplets shown in red, they look pretty much the same at 12 hours, but after longer times on a poor diet, they start changing. So, and what we saw with this volcano plot where the y-axis shows the p-value of the fold changes and of the differences, and the poor versus rich difference in expression is shown on the x-axis. So what we see is that in this quadrant here on the upper left is the down-regulated proteins on a poor diet. And on this right upper quadrant here is the proteins that are upregulated on a poor diet when you do that switch from rich to poor. But another way, simpler way to think about this is that the ones shown in red are enriched when the flies are on a rich diet and lower on a poor diet, and the ones that are shown in blue are enriched on a poor diet and lower on a rich diet. And a lot of those enzymes that we identified here, out of 2,500 proteins, 450 were changing, and the majority of those were actually metabolic enzymes. And we looked at just some of the metabolic processes that were being affected. And there was, for example, energy metabolism was being affected. And what we were seeing was that on a rich diet, the glycolysis and enzymes involved in like pyruvate and acetyl-CoA production were at higher levels, but the TCA cycle and oxidative phosphorylation enzymes were at lower levels. So this part of the pathway was being activated, but not for production of ATP. That would be one interpretation. And instead, it was being used for building blocks, for example, or maybe for production of acetyl-CoA that could change the epigenetics. We don't know. And then another example, there was an increase in fatty acid oxidation, maybe to produce ATP from this source, since it's not being produced from this, but we don't know, or it could be for production of ketone bodies, which could either signal to other tissues or be transported as a source of energy. And another example was increase in phosphatidylethanolamin synthesis, which has roles in lipogenesis and also signaling roles. And so, but there was like so much information, we decided to just test these 3 pathways, and I'll just show you what we did for the glucose metabolism and shift to pyruvate acetyl-CoA. What we did is these enzymes shown in red are all the ones that are coordinately up on a rich diet, but the ones with a dark box are the regulatory enzymes. So when we looked at the effects of knocking down those enzymes in the ovary, just to see if it made sense that they could be relevant to the ovary and how it responds to diet, we chose those regulatory enzymes. This is just a close-up of the same enzymes. And if we look at the germarea here, what we see, this is a control RNAi, and this is, you don't see any death, which is shown with a red marker here. against a cleaved caspase. But if you look at the knockdown of HEC-C, which controls this step here, or PEPC-K, which controls this step, which all leads to production of pyruvate. If you knock down either of those enzymes just in the adipocytes, you see increased death of early germline cysts, and the germaria are significantly smaller. And when we quantified this, we saw that with 2 different PEPC-K RNAis and the HEC-C, there was a significant increase in the death, in the percentage of germaria that had dying cysts. And although in this first experiment, knocking down these 2 enzymes individually did not have a phenotype, after the paper was published, we knocked down both enzymes together and it had the same exact phenotype. And this was very specific because there was no effect on stem cells, no effect on later stages of oogenesis. But we don't know exactly how the signal, the change in metabolic state of the fat cells was communicated to the ovary. But just to summarize, a little bit of what we found in terms of how the adipocytes communicate to the ovary, then we showed insulin signaling is sensed, amino acids are sensed, metabolic enzymes that change with diet. If you alter them, like experimentally, they have effects on the ovary. So, and this is just the tip of the iceberg for what adipocytes can be doing. So, so we decided when a new postdoc joined the lab, we decided, okay, let's try to get away a little bit from adipocytes and not have a preconceived notion of where signals might be coming from, and instead just use nuclear receptors as an entry point to try to understand, because nuclear receptors are essentially transcriptional factors, so they can have huge effects on a cell's function and fate, but they have a ligand binding domain that makes them be regulatable by a huge variety of signals, many of which can either come directly from the environment or be in your circulation after production in some organ or anything like that. So we thought, well, if we look at what roles these nuclear hormones have throughout the body to control oogenesis, we can understand better how they are participating in this whole body physiology regulation of the ovary. So Lassie came to the lab, and she actually started her lab, like some of you, right in the middle of the pandemic, about 1.5 years ago at Indiana University in Bloomington. So she's featured here because she's the last postdoc to graduate or leave the lab. So Lassie decided to, again, because of what I just said, for nuclear receptors and all the different roles that they are known to have, she was interested in how they would factor into oogenesis. But beforehand, like including work from our own lab and work from other labs, there were a fair number of nuclear receptors in flies, and they all have mammalian homologs that are listed here, that have roles in the ovary, but they all had roles that were intrinsic to the ovary. And the only one that had been examined outside the ovary for how it could help, in how the organs interact to refine the response was the ecdysone receptor, which, in addition to having multiple direct roles in the ovary that we found and other labs found, Alan Spreading's lab had found that Matt Sieber in Alan Spreading's lab had found that the ecdysone receptor also functioned in the brain to control feeding behavior, so that females would eat more when they were stimulating oogenesis. But we wondered, like, how many other nuclear receptors could have roles in other organs to then crosstalk to the ovary and help refine the response besides ecdysone receptor. So she took an approach where she used a ubiquitous driver for RNAi that worked in all somatic cells, but for technical reasons, not in the germline. And she knocked down all the different nuclear receptors in flies. There's about 20 of them. And she did that in an adult specific way because we didn't want to get into developmental problems. And she started out ubiquitously because we didn't want to be biased towards any one tissue. And she focused on 2 candidates from this screen to characterize, both in my lab and then later in her lab. So one of them is 7-Up and the other one is HR4. And I will not talk about HR4 today, but she did have a recent paper on that, and she's taking both of these projects into her lab. So when she knocked down, for example, 7-Up and counted the number of eggs per day, the controls are in gray here, and 2 different 7-Up RNAi lines are in pink or whatever these colors are. And you can see 5, 10, 15 days, there's a specific significant reduction in the number of eggs that the females lay. And I don't have time to show all her data, but I'll just show you like what steps of oogenesis were affected by ubiquitous RNAi, and then she actually figured out which organ was responsible, but I'll just summarize that last data. But beforehand, I'll just tell you, 7-Up in flies is involved in eye development, neuroblast specification, and the CoopTFs in mammals also have roles in the nervous system, similar to the flies, but they can also be abnormally regulated in tumors, and they have roles in angiogenesis and even in control of luteinizing hormone in mammals. So Lassie tested all steps of oogenesis, but I'm only going to show you the ones that are affected by 7-Up. So, and this finally the promised picture of the stem cells. So this is a control knockdown. The germline cells are labeled with VASA here in red, and the germline stem cells can be recognized by location and other markers that I won't go into details, and they're up against the niche here. And in one of the 7-Ups knockdown that's shown here, you can see 1 stem cell, and there's fewer germ cells here. And DAP is showing all the nuclei of all the cells around. So when Lassie counted the average number of stem cells over days of RNAi, starting adult specifically, if you look at this dark, I think it's gray, there is a small reduction in stem cell number from the start. But if you focus on the slope of the other curves, you can see the rate of loss is faster when 7-Up is knocked down. But there are no effects on proliferation or the size of the niche. The other step that was controlled by 7-Up, if you compare the luciferase RNAi control versus a germarium from the 7-Up ubiquitous RNAi, and here we're looking at another marker of cell death, which is like a tunnel-based assay, and the dying cells are shown in green. And this is an example of an early dying cyst in the germarium. And what you can see, she quantified at 0 and 10 days the number of dying cysts in the very anterior region here, near the stem cells, and in the more slightly more posterior region where the 16 cell cysts are getting formed. And what you can see is that at 10 days of RNAi, there's a small increase in death of cysts in this most anterior portion, but when they get closer to being enveloped by follicle cells, there's a really drastic increase in the percentage of germaria that show these dying cells when 7-Up is knocked down. And the third step of oogenesis that was affected by 7-Up knockdown in the fat cells was vitellogenesis. So this is a control ovariole. Now, instead of showing you just the germarium, I'm showing you the entire ovariole, and you can see the developing egg chambers with DAPI labeling their nuclei. This is a big vitellogenic egg chamber. This is the size of the oocyte here. But in 7-Up knockdown, you can see that there is a higher frequency well, this example shows a degenerating vitellogenic follicle here with the pycnotic nuclei. And if you look at the graph, there is a significantly higher percentage of ovarials that have this dying vitellogenic follicles if 7-up is knocked down in adipocytes. Sorry, I'm spilling the beans. If 7-up is knocked down in throughout the fly. So then Leslie set out to try to figure out what specific tissue is required for each of the 3 effects of a 7-up on the ovary. And what she found, and I don't have time to show the data with the specific drivers, is that we weren't able to avoid the adipocytes. So it turns out that the adipocytes were the cell type where 7-up is required in order for its role for maintaining stem cell numbers and promoting survival of early cysts. So if she knocked down 7-up in adipocytes specifically, there was the stem cell loss and increased cyst death, just like we saw for the ubiquitous knockdown. And it turns out that for the vitellogenesis, there was no role in the adipocytes, but if the 7-up was knocked down in inocytes, which have the liver-like function, then vitellogenesis was affected, but not the other processes. So there's this interesting separation of how the 2 parts of the fat body are cooperating to control oogenesis, but through different processes. And so this brings up a lot of different questions, like what are the inputs upstream, because 7-up is an orphan receptor, and questions about how similar this is for other nuclear receptors or for the mammalian homologs. But another kind of obvious question is like, what are the targets of 7-up in the fat body? Like, what are the targets in the adipocytes, inocytes, or in the fat body? And how are they mediating these effects on the ovary? And my lab, like the last paper that Leslie had from my lab was an RNA-seq study where she knocked down 7-up using both the inocyte and adipocyte driver simultaneously. So she was knocking it down in the entire fat body. She manually dissected the fat body or away from the rest of the fly. And she did RNA-seq, control versus 7-up. And even though we kind of expected to see a lot of metabolic genes because so many nuclear receptors are important for metabolism and because of the fat body is such a metabolic organ, what she actually found, there was no metabolic gene regulated by 7-up. And instead it was controlling immunity and xenobiotic response genes. And so now her lab, she's going to try to understand, how these factors might be affecting oogenesis and what are the upstream inputs that might trigger changes in 7-up function and all that. But that's her adventure for her lab. And finally, I know I'm running out of time, but I just want to briefly describe the project of one of my current postdocs, and our paper on this is currently in revision, which is a project looking at a different type of environmental input from diet, which is how does temperature affect oogenesis? And there's obvious reasons to be worried about temperature nowadays, because of all the consequences of global warming and turning all the weather patterns crazy throughout the whole world. And that also leads to habitat loss, which makes it even more challenging for cold-blooded organisms to regulate their temperature, like with all these compound effects. And insects, of course, have so many species and contribute so much to biodiversity, but they also have important roles in food production as disease vectors and as beneficial insects for agriculture, or they can be pests in agriculture. I hope people can still hear me because I'm frozen. Okay. We decided to look at this in flies, but we didn't want to look at like ridiculously high or ridiculously low temperature. We thought, okay, people who work with flies normally treat 25 degrees as the ideal temperature in the lab, but we often work with 18 degrees, 29 degrees to manipulate our tools or to maintain our stocks, etc. So we wondered, how are these like basic temperatures that we use in the lab affecting the ovary? And if we look at 25 degrees here in black, you have a nice curve of egg production, which kind of goes down as the females get a little bit older. But if you put the flies at either 29 degrees or 18 degrees, there's a reduction in the number of eggs produced. So in terms of, in nature, I think the faster you produce your eggs, the better, but as long as they're good quality. So the question was we didn't expect the mechanisms to be the same, but what are the mechanisms, the cellular mechanisms in the ovary that are responsible for this decrease in egg production under this, let's call them suboptimal temperatures in the lab. So, and I'm just going to show you for each temperature, because Anna, my postdoc, looked at every process we could think of in the ovary, but I'm just showing you the ones that are actually affected. So at high temperature, the 29 degrees, if you compare, actually this early germline cyst is actually affected both in high temperature and in lower temperature. And this is, again, another example of germaria with the APOP tag, the tunnel labeling assay for cell death. And what she could see was that if you compare to the 25 degrees control at 5 days or 15 days, there is a marked decrease in the percentage of germaria with dying germline cysts at 18 degrees. And there's an increase in the percentage of germaria with dying egg chambers. With dying germline cysts, I misspoke, inside the germarium. So the survival of cysts is improved with lower temperatures, and it gradually, the death increases with higher temperatures. And that's a really significant proportion here after 15 days. So that's one step that's temperature dependent. And vitellogenesis was not really affected significantly at 18 degrees, but at 29 degrees, there was especially at later time points, 15 and 20, there was a very large and significant increase in death of vitellogenic egg chambers at 29 degrees. So from warm temperature, the reason why the egg production is reduced is because of a negative effect of high temperature on early cyst survival and on the survival of vitellogenic egg chambers. And at cold temperature, we saw that improved in cyst survival, no difference in vitellogenesis, but the improved cyst survival wouldn't explain lower egg production. So what we did is like a lineage tracing system based on FLIP-FRT that I won't go into details, but at time 0, only cells that are mitotically dividing can be labeled through this lineage tracing system. And in the case of the germline, they only divide mitotically while until they make the 16-cell cyst, and then the mode of cell cycle changes. So we can label cells at time 0 that were in this very early stage here in the germarium, and then see how fast those labeled cysts progressed over time at different temperatures as a measure of the relative growth rate. And this is just an example of what the data would look like. These are ovarioles at different temperatures. The labeled cells were induced at time 0, and then the label is retained by the progeny of the cell and the cell, and is recognized by beta-gal antibodies. And you can see at 18 degrees, this is like the farthest in this ovariole that the clones have progressed. You see multiple labeled cells, and this is how far they progressed. At 25 degrees is another example, 29 degrees. But we quantified this, And when we see the y-axis is not really a real number, it's the positions anterior to posterior along the germarium and how far those cysts have developed. And these are 1, 2, 3, and 4 days after induction of the label in this anterior-most region. And you can see that between 25 in black or 29 in red, there is not really much difference in the rate of growth and development of the cysts. But at 18 degrees, you can see that they are really lagging behind because oogenesis is slowed down. The growth of the follicle, the development is slowed down. So this then tells us that at cold temperature, the main reason for reduction of egg production is a reduction in follicle growth. But we don't know the mechanisms. It could be a regulatory mechanism, it could be thermodynamic differences, or it could be a slowdown in aging of the organism. So we don't know at this point. But the number of eggs that flies produce is only part of the equation of whether the flies are fertile, because if those eggs are of poor quality or they don't get fertilized because the males can't fertilize it at high temperature, then you don't get an embryo, and that's no good in terms of trying to perpetuate the species, right? So we also looked at the quality of the oocytes and also at and male fertility, but I won't go into the male fertility stuff. Suffice it to say, the males become completely sterile really fast at 29 degrees. But we tested the females specifically by looking at the readout at using fresh males that were not incubated at 29 for very long, to see if the embryos that these females produce, like the oocytes they made, could support embryo development and give rise to a larva. So this is a percentage of hatched eggs. And if we take either mated females that were maintained at either 25 degrees or 29 degrees until they were 20 days old. So this is already reducing the hatching rate even at 25 degrees because the females are older. But if you compare to 29, it's a much lower rate of hatching rate. And even if you take virgin females, which haven't mated at all for most of this time, and then you add the males at the very end of the experiment, you can see that at 25 degrees, there was a much higher success rate of embryo development because the oocytes maintain their quality better than at 29 degrees. And interestingly, we decided to test known warm temperature sensors that are well characterized for mediating behavior changes in response to changes in temperature in the flies. And we thought, well, we should at least rule them in or out as part of this mechanism. And we looked at the hatching rates. This time is at 10 days. That's why they are a little bit different. But if you look at 10 days at 29 degrees, in the control, you see this reduced hatching rates. And in the TRPA1 homozygous mutant, there is an improvement in the hatching rates, suggesting that the mutation of this warm temperature receptor can improve the oocyte quality at 29 degrees. And but the other temperature sensor did not seem to have any role. But one experiment that we are doing now, which the reviewers of the paper asked for, and I think it was very fair, we are doing a rescue experiment with a TRPA1 genomic rescue. And we are also in the process, which the reviewers didn't ask for, but I also thought, yeah, we should do that. Make sure that this effect is really temperature specific and is not some other role of TRPA1 independent of temperature that just improves oocyte quality. So we have to strengthen this data some more. But to summarize, this is the current state of our interpretation of our data, that at cold temperature, I didn't show you those data, but germline stem cells are maintained better, cysts survive better, oocyte quality is better, which I didn't show you that, but the oocyte quality, the hatching rate stays really high all the way to 20 days, but the follicle growth is slowed down. And at warm temperatures, there's no difference in follicle growth, and the early germline cysts die more frequently, vitellogenesis is impaired, like the vitellogenic stages die, and the oocyte quality is drastically decreased, and somehow TRPA1 seems to be playing a role in that. So with that, I apologize for my technical troubles and for going late. But this is just the most important slide to thank all the past and current members of the lab. I already mentioned some of them, but these are other current members of my lab. There's 2 postdocs, Ana and Rodrigo, 2 graduate students, Emily and Rehan, and we have 2 master's students in the lab for a year. And we have a high school student that comes in the lab, you know, a few hours, solid number of hours in every week. And she's super excited about science too. And thank our sources of funding, the super generous flight community. And if you guys have any questions, I'll be happy to answer, although everybody must be tired with my long seminar, but I will stop sharing and to give you guys a chance to.
Thanks so much, Daniela, for a fascinating talk.
Yeah.
Any questions from the audience?
Can I ask the question? So I'm kind of interested in 7-Up story. That is the 7-Up is mediating the metabolic changes, do you think, or as a signal? What kind of signal can regulate? Do you?
We thought it would be acting to control metabolism because we had already linked the metabolism in the fat cells with the ovary. And because so many nuclear receptors really have major roles in controlling metabolism. But then when Leslie did the RNA-seq, there was like no changes in any metabolic gene.
I see. Is that
So
possible that
that could be like, you could think of a really like more indirect mechanism where those changes in immunity and xenobiotic response genes could lead to like a translational regulation of metabolism, but definitely not transcriptionally.
Okay.
There's no evidence of transcription.
How about the metabolic status changing the expression of 7-Up or localization of 7-Up from the cytosol to the nucleus? Is that?
We haven't tested that, but we felt like because we identified the targets, they seem to be pointing in other directions. So I think Leslie is just going to focus on, I mean, regardless of whether, because now this brings like 2 questions, right? Because the fact that all this immunity and xenobiotic response genes are altered in response to 7-Up brings up the question, maybe that's how 7-Up affects the ovary. But we don't even know like how immunity genes or how xenobiotic response genes affect the ovary. So it's almost like Leslie, I think I'm not gonna meddle, like try to like say any, like my impression is that she's gonna like not just think about them in terms of 7-Up targets, but as its own question, right? How does changes in immunity genes, or how do changes in immunity genes and xenobiotic response, how does that affect the ovary? And if she understands that, she can try to see if that is plausibly the mechanism, how 7-Up is affecting the ovary. But in a way, she's in a good position because it almost doesn't matter if those are the relevant targets, because it gave her an idea to figure out a whole new angle for how physiology is interacting with the ovary, right? So, and she's super excited. It was
right. Thank you. Interesting.
I see a question in the chat. How evolutionarily conserved do you expect these regulatory mechanisms to be? I think that usually the specifics might not be super conserved, but the general principle and the basic signaling pathways tend to be conserved. So, for example, after that would apply to all of the above that I talked about. But for example, when I looked at when I started looking at the effects of diet and insulin signaling in the early days, right, in my lab, and like starting with my postdoc, there wasn't much out there, but then afterwards, like at least the fly people started paying attention to that. And then they started testing whether these responses were occurring in the testes of flies or in the stem cells there, or in the stem cells in the intestine. And there are like not identical rows, but parallel rows of insulin signaling in the testes, although, you know, the cell types where it might be required might be a little different. Like in the testes, insulin signaling intrinsically can control the maintenance of stem cells, but in the ovary, it's all through the niche. So there might be details that are different. Or in the intestinal stem cells, there are roles of insulin signaling, but the mechanisms are different, and there might be a local source of insulin for that. And in mammalian systems, it's a lot harder to address because you can't easily pinpoint to the stem cells. But if you look at, and we wrote a review about it in 2012, so it's already way out of date, but even at that time, there's like tons of indications that at least diet-dependent pathways or pathways that are tied to the physiology can have all kinds of effects on different mammalian stem cell systems. So yeah, so I think that not identical mechanisms, but similar or general principles or like the relevant pathways, there's going to be a lot in common across many organisms. I hope that answers the question.
Daniela, I had a question. So you're delineating, in a sense, checkpoints, right? So through this process that are sensitive to the environment and nutrients. And one of the key decisions is for the germ cell, the stem cell to go into the mitogenic, or essentially the pathway where they start to undergo the incomplete divisions, where they're committed to becoming eggs at some point, right after the mitosis, incomplete mitosis. Do you know much about whether that specific step is regulated and how that might work out, or any expectations that how that would be regulated at the molecular level?
Yeah, so in terms of, well, so we know from mutants from other labs that there are ways to block differentiation that can block either at a really early stage before the daughter of the stem cell really commits to being a cystoblast, because there is a marker that comes on in cystoblasts called BAM, and that's really important for differentiation. And that's work from Ting Zi's lab and from Dennis McKeerin lab when he had his lab. And so BAM is an important differentiation factor. And DPP, BMP signals are produced by the niche, and they actively repress, the signaling pathway responds in the germline stem cells, and they actively repress BAM expression in the stem cells. So it's a transcriptional step of regulation. And then once BAM is de-repressed, you can go on and differentiate, except that it's not so simple because it's biology. So there's multiple translational regulators that crosstalk and further reinforce that response. There's additional mechanisms that make sure that DPP signaling is really turned off very fast once the cells move away from the niche. So there's multiple additional mechanisms just to make it really, really robust, that when the cells decide that they're going to become a cystoblast and differentiate, that really moves on. But there is another gene, it has a cute name too. I forget the name, but it's actually a gap junction protein that is link, not our work either, but it's linking the germline stem cell to the niche. But if you mutate that gap junction, the stem cells divide, they accumulate as if it was a cystoblast tumor, but they don't turn on BAM. So it's almost like there is another step right before a pre-cystoblast, maybe right before they turn on BAM. So there's like genetically, you can separate these steps. Now, as far as like our work, we haven't connected our work with the specific differentiation factors, but we did connect, in the case of the insulin signaling, we know that the insulins come from the fat cells and they act directly on the germline stem cell to reduce how fast they produce cystoblasts. But that's not a fate decision, it's just a cell cycle, how fast they divide. And that's all at the level of G2. And we know it's through the foxo and not through TOR, like not through TOR downstream of insulin signaling, but TOR has a separate role independent of insulin signaling in also controlling G2. It seems that is we found that the germline stem cells have a really, really long G2 phase to begin with. They have an almost non-existent G1, and they have really weird expression of cyclin E, because cyclin E is normally on in late G1 and like S, but in the stem cells, it's on in G2. So in mitosis. So it's almost like they are preparing for G1 before they even finish the previous division. And then they barely have a G1 and they are ready to replicate DNA again. And it turns out that's true in embryonic stem cells. And it's true like, and it's like, it's true for the germline stem cell, but when you get to the early cysts, they just have normal mitosis with no weirdness with G2 or these markers. And one of my postdocs, the same one who did the Dyson work that I did not mention, she has her own lab at East Carolina University, and she did a really cute study looking really carefully at how all the cell cycles take place using lots of markers in the early ovary. But in the case of the maintenance of the stem cells, it turns out, I don't know if you remember the introduction I mentioned, the stem cells are attached to the niche, right? Because it doesn't do any good to have all this like special signals near the niche if the stem cell is just going to move away from it, right? So E-cadherin is super important for attachment. And Ting Z showed that if you mutate E-cadherin, you get stem cell loss. So what we found was insulin signaling insulin-like peptides act on cap cells, and they do two separate things on cap cells that we found. One was that it controls the levels of Notch signaling, and Notch signaling is important for maintaining the number of cap cells in the niche. So that was how the size of the niche is controlled by insulin. On top of that, we found that adhesion to the niche is also controlled by insulin, because insulin controls the level of E-cadherin between the cap cell and the germline stem cell. So it's like enhances the Velcro there. So those are kind of mechanisms that we found in for TOR signaling. We found a role for TOR signaling maintenance. And then another lab published shortly after us similar findings, except they focus more on hyperactivity of TOR, because either one messes up the maintenance. And it turns out in that case, there was deregulation of BAM. You get premature expression of BAM, and then the cells differentiate. And we also found in the case of Dyson, Elizabeth, that same postdoc that did the cell cycle study that I mentioned, she found that when she was a postdoc in my lab, that Dyson was controlling maintenance of stem cells, but it was through some sort of interaction with the ICY chromatin modifiers. So it's like there's lots of different mechanisms at play that we found so far, but it's a little bit harder. And we know there's mechanisms that are independent of BAM and all that, because for some of the stem cell loss that we get because of physiology, for example, the manipulation of the amino acid transporters that cause the stem cell loss, there's no difference in the levels of BMP signaling or of E-cadherin. So it has to be some other reason, but we don't know. Sorry, that was a very long-winded response.
Great. Yeah, thanks so much.
I talk too much. You guys might have noticed already.
Great. Any more pressing questions? Otherwise, let's thank Dr. Drummond Barbosa one more time. And thanks so much for your seminar.
Yeah,
And
thank you.
thanks everybody for being here.
Yeah, thank you so much for having me over.
Thank you so much.
Yeah, I had a great time, and sorry, my seminar had some problems and I talked too much, but I hope it still made sense. Yeah.
It was fun. Thank you.
Okay,
Thanks, everybody.
thank you.
Thanks, Daniela.
See
Bye,
you guys
Daniela.
later.
Bye.
Bye.